yeah, thank you for asking me along to talk. So yeah, I will talk briefly on each of those projects that Stuart mentioned. So what I do, I'm a biological anthropologist, also known as an osteoarchaeologist. So I essentially analyse human remains that come from archaeological sites or historical contexts, and I look at the evidence on the skeleton that can tell us something about both those individuals, and then also look at populations as a whole. So if we're lucky enough to have cemetery populations, then we can kind of extrapolate that out and think about populations as a whole. So I came to this, my background was originally in archaeology. So I started off as an archaeologist. I then moved into academia. So I did my PhD at the University of Bristol. I worked in a forensic science department for a while as well. And I'll talk about where I use some of those kind of techniques as well. And now, as I said, I'm at University of Winchester. Yeah, just to flag up, so just in case you weren't expecting to come to a talk about human remains, I've just put a note on there. Please note the talk does contain images of human remains right from the off. So just in case anyone is sensitive to that, you can jump out now. Okay, so as I've mentioned, what I do is I work in osteoarchaeology. So, and specifically for me, that's looking at human remains. So that's looking at the skeletal remains of past populations. So we tend to start off with these remains coming from excavation. And of course, in the kind of current climate in the UK, a lot of these remains come from excavation in advance of development. So most of the skeletal collections I work with are excavated because the area is being developed for one reason or another. The image here on this slide, this is at the Priory, Taunton Priory in Somerset. So this is myself excavating one of those skeletons, and at that site, this is a very large medieval cemetery, of which the skeletons were excavated that would have been disturbed or damaged by some new flats and some new houses that were being developed in this area. So we tend to start with the excavation phase. Then once the remains have been uncovered and excavated, we record everything really thoroughly. So for example, burial position, we kind of draw everything, photograph everything, and then carefully package everything up. That will then go back to the lab to be looked at in detail by the human remains specialist. So we move from the excavation to the analysis phase. And here we have an image in the left. So this is one of the skeletons actually from the 19th century Bristol cemeteries that I'm also looking at. So this is what we would do. We would lay them out in the laboratory in anatomical position. And then essentially I would look at every bone in the body. I would aim to work out things like their sex, are they male or female? How old were they when they died? I would take measurements to try and determine their estimated stature. Look for human variation, so look for interesting features. Although anatomically our skeletons are kind of pretty much all the same, there's a lot of variation in shape and morphology. Some of us have a few extra bones than other people, so we look for all that variation. And then another important thing that I do that I'll talk a bit about as well is looking for evidence of disease. So what we call paleopathology. So looking for disease that we can see on past people and then on past populations as well. So we would then gather all that information, and then of course we would move on to the interpretation stage. So I've just put an image here. So this is kind of a posed Victorian photograph of a sick room image. And I've put that image up because the skeleton that is laid out in the lab here, they had evidence for tuberculosis on the skeleton. And I will talk about that a bit further on as well. And so again, we can then think about, what does that mean for this person while they were alive? So although I'm dealing with individuals who are obviously a long time deceased, we like to think about what the evidence we see on their bodies, so what that evidence for disease, how that may have affected their lives. So, you know, everything we look at tells us something about that individual's life. So we therefore, in this case, might interpret this was a young individual, so they were aged to kind of 12 to 15 years of age, so they're kind of adolescent. So, you know, they may have been being cared for kind of at home and being kind of looked after on their sick bed, as it were. So that's the kind of stage we move through. So from the remains being excavated through to that analysis and then moving on to that interpretation stage. So I was just going to show you some of the methods just to start off that we use. So, you know, how do I know whether I'm dealing with a male or a female? So how do I work out things like sex determination? So I've got some images here. So we've got an image of a female and a male pelvis. The pelvis is the best bone in the body for sex determination, for obvious reasons, that females have to have pelvises that are wide enough to allow childbirth, whereas obviously males don't have to go through that. So I've put an arrow looking at an area towards the back of the pelvis, which is called the sciatic notch. And this is wider in females than we see in males. So hopefully you can see that on that image, that the one on the left is wider than the one on the right. The other image I've put down is of a female pelvis. And again, we're looking at the angle now at the front of the pelvis. And again, that's showing this really quite nicely wide arch. In a male, it would be narrower. So again, we're looking at these angles that are related to the width of the pelvic girdle. So that's really useful if we've got the pelvis. If we don't have the pelvic bone, we can actually also look at the skull of an individual as well. So again, here I've got examples of a male and a female skull. And then I've also got a male skull illustrated on the right-hand side, picking up some features. So when we're looking at features of the skull, we tend to look for features that males, their skulls tend to change as they kind of grow into adulthood, and also as they get older. They tend to get what we call more robust figures, more robust morphology. So particularly in the area of the eye orbits, males tend to have this supraorbital area, so this area by the eyebrows tends to be more prominent. They also tend to have foreheads that slope further backwards, whereas females have quite flat, high foreheads. If you're sitting next to someone, you can kind of, you know, look at your friend next to you and kind of compare. The other feature that I've got pointed out with an arrow here is called the mastoid process, and this is the lumpy bit just behind your ear that you can probably feel if you feel just behind your ear. So that's for muscle attachments. And again, in males, that tends to be larger than we see in females. And the other thing that's pointed out, although for me, I've got people's images over the slide slightly, but there's also an arrow looking at the back of the skull, so the occipital bone. There's an area called the nuchal crest, and this is where, again, the muscle attachments for the neck attach. And again, that's much more prominent on males than we see on females. And these areas, we would score them all on a scale, kind of using 1 to 5, with 1 and 2 being more gracile and female, 4 and 5 being more robust and male. And of course, sometimes we do have individuals that kind of fall in what we call an intermediate level. So we're essentially scoring on this gradation scale between hyperfeminine up towards hypermasculine. Of course, we can only do this with adults. So those sexually dimorphic characteristics, so those differences that we see between males and females, only really appear kind of once somebody's moved through adolescence. So we can't sex immature individuals using these techniques, but they're very accurate for looking at adults. So that's how we can kind of come up with sex determination. There are other areas, so even if we don't have the pelvis or the skull, we can look at the long bones, so we can kind of take measurements, which can help us, particularly if we know something about the size of the population that we're dealing with. Bones like the sacrum we can also use. We can use the lower jaw as well, which also has more robust features on males than we see on females. So then thinking about age determination. So whereas with adults, it's quite straightforward sex determination on adults, but we can't really do that with children, with immature individuals. When we're looking at age, it's actually easier. We can be more accurate when we're looking at children and developing individuals. And that's because they are developing and growing. We know the kind of timings of things like dental eruption. So we have the deciduous dentition, that first dentition that children get. Then those teeth get pushed out by the adult dentition, that permanent dentition coming up. And again, we know the timings of these things really well. We can use modern dental x-rays to look at children of different stages to know those timings. So with immature individuals, aging is quite straightforward. But with adults, it's much more difficult. And that's because we're not looking at developmental stages any longer. We're actually looking at degeneration. So sadly, once we kind of reach adulthood, it's here when we start to kind of degenerate, our skeletons start to degenerate. So what I've put on this slide here, this is just one of many various areas that we can look at across the body, looking at the level of degeneration that we see. So I've picked the pelvis again. So again, the pelvis is really useful for age determination as well as sex determination. And here we're looking at the articular surface where the two bones of the pelvis meet the sacrum, so the base of the vertebral column. So it's an articular area, but it's one of those areas that doesn't really move. So it's not like an elbow joint that you kind of move around and if you do a lot of heavy lifting, it would have an effect on it. It's a joint that doesn't really move depending on your lifestyle. So therefore it's quite stable across all different individuals. Occupation won't play a factor in the aging process for it. And so that's why we use it. And we tend to use these kind of what we call non-movable joints when we're looking at aging. So again, I've just put some arrows on the images. So our example on the left-hand side is of a young individual. So someone kind of 25 to 35 years of age. And then the image on the right is of a more elderly female of kind of 60 plus years. So in the young individual, the bone has these kind of little lines running across it, these striations, which is what we see in young bone. Whereas in contrast, in the older bone, There's lots of porosity, so there's lots of quite large holes. The bone is not as fine-grained anymore, it's much more dense. And we also see where the second arrow is, a little projection. So you start to get these little bony projections as well that occur. So these are all features of degeneration that occur with age. So they help us to determine where this individual falls. As you can see, I've kind of got a 10-year age range, even for that younger individual. So as I've mentioned, it's not as easy to kind of give a specific kind of age to adults. So we tend to place them within age range categories to try and be certain that we're getting them into the correct category. So there's a couple of areas on the pelvis we can use. We can also use the joints in the skull. So the skull has lots of suture joints where all the different bones come together. We can use those as well. For aging adults, we can also look at the dentition, but this time we're not looking at development. We're actually looking at the wear that we see on the dentition. But of course, if we look at that, we need to know something about the diet that the population is eating. Okay, so I just focused on kind of a bit of sex determination, a bit of age death determination, because that was quite important for my first project that I'm going to talk about. So this is the collection of remains that are within the mortuary chests, which are held at Winchester Cathedral. So Winchester Cathedral was creating a new exhibition. They were having some conservation work done on the mortuary chests. So it seemed like a good time for them to actually, because those chests were coming down, they thought it's a good time to have a look at the contents of these chests. So the contents of those chests have allegedly been in there for kind of several hundred years. They are a commingled collection. So that means there isn't like separate skeletons placed within the chests. There's just a big mix of lots of different bones from the skeleton. So part of the role that I was playing, so I was essentially taking all the remains out of the chests, recording them as if it was an archaeological excavation. So photographing everything, numbering every bone, drawing every bone within those mortuary chests. So essentially excavating the chests. And then obviously looking to try and obtain biographical information for each bone. So although all these bones were separate rather than being individuals, thinking which of those bones can I try and sex? Which of those bones can I try and assign an age at death to? And then also utilising more forensic techniques. So again, obviously in forensic cases, they may come across commingled remains. So there are various techniques that they use to then try and reassociate body parts back to an individual. So some of these are utilising measurements and using statistics to try and work out if different bones could potentially belong to the same individual. So with these remains in the mortuary chests, there are a list of different names of individuals that are thought to potentially be contained within them. So when the new cathedral was built, Old Minster, which stood kind of on the area where the cathedral was, that was knocked down. They then exhumed some of the good and the great that were buried around the altar of Old Minster. So they then translated those remains, they took them into the new cathedral, probably as individual skeletons, maybe at that time, depending how well they knew where everybody was placed. And it seems they were held in kind of lead caskets for some time. but they have, you know, they've moved around various places in the cathedral. Historical documents show that they've kind of made journeys to various different parts of the cathedral. And they've been in various different lead caskets and wooden mortuary chests as well. So this slide here, so this just shows some of those individuals that are thought to have been placed within these mortuary chests, ranging from the 7th century up until the 12th century. So various kings, one queen, Queen Emma, and some bishops as well are kind of thought to be within there. And the image, that's just one of those chests as they sit up on the presbytery above the choir. So what I did then, so I kind of went through this process, first of all, looking at each bone individually, trying to work out how much information I could get about that individual. Then utilising these techniques, some of them essentially just laying out, like, you know, how many leg bones have I got, how many femurs, how many thigh bones, laying them all out next to each other, working out my lefts and my rights, and then essentially pairing up which ones look like a match. So that's kind of a first kind of process, and then working using measurements and statistics to then try and work out which joints could potentially fit together and be a match. So there are certainly more individuals within those mortuary chests than are named on that list that I showed you. And there was also, as well as being adults in there, there were also some immature remains as well, which was quite interesting. And I probably found those the most interesting personally, having worked quite a lot on juvenile remains kind of previously to this project. So some of the work that we've done has gone into this new exhibition at Winchester Cathedral, which opened in 2019, summer 2019. So as well as kind of the morphological assessment that I do, there have been various scientific techniques that were undertaken to try and, first of all, determine, are these remains actually potentially what they say they are, or could they be kind of later additions? So we did some radiocarbon dating, so both with the Oxford University and University of Bristol as well, did some sampling and radiocarbon dating. And the dating did come out within that period. So we did have dates as early as the 7th century and as late as the 12th century, but you know, everything kind of fell, all the dates that we took fell within that period. So that was really good. So that seemed to work. We've also done some work with stable isotopes. So particularly this is looking at nitrogen isotopes, which can potentially tell us something about diet. So nitrogen gives us an idea of how high an animal is up the trophic level. So essentially how much meat they are eating. And again, quite important in this case, how much fish, how much marine foods individuals are eating as well. The other stable isotope in particular we are looking at is strontium. So rather than tell us something about diet, with strontium, it tells us something about geographical location where an individual grew up. It's like a signature of the geology that you intake with your food and water. And it places a signature in the enamel of your teeth. And that's placed in there as you're growing. So as your enamel develops, it's like a signature from your childhood. So that can be useful in trying to work out where individuals actually grew up. Was it in the Winchester area or was it kind of somewhere else? So that's some of the scientific techniques that we've been using on these remains. So just to quickly mention a couple of the individuals that made it into the exhibition. I mentioned that we found some juveniles, which obviously we didn't expect. They were actually ones that I suppose I actually started looking at those. To me, they were the easiest ones to work out full skeletons for, because they were immature. They were quite easy to kind of work out which bones belonged with which skeleton, whereas a lot of the bones in those boxes were all from males of around a similar sort of age, so it made it much harder to try and separate them into individuals. So we have one of these individuals was placed into the exhibition, and a facial reconstruction was undertaken by Richard Neve, who's a kind of well-renowned forensic reconstruction artist. There has also been some initial ancient DNA analysis, and in this case it was used to tell us that that individual was actually a male. And in fact, there are two juveniles around 12 to 15 years of age, and they are both males. So at the moment, I've kind of got who could this be with a question mark. Professor Barbara York at University of Winchester has some ideas that they may be relations of William the Conqueror. There were a couple of young princes that were killed in the New Forest, and they have a kind of relationship to Winchester. So, but work is ongoing, kind of thinking about who these individuals might be. Our other individual that we have, which was slightly easier to work out as a separate individual, because this was the only female that we appeared to have from all the bones within the mortuary chests. So the only female we have named is that of Queen Emma. So it's possible that this could be Queen Emma's remains. So from my kind of look at the bones from the osteoarchaeological information, she is kind of over the age of 50. So Emma died in her kind of late 60s, so that fits quite well. If we look at the carbon-14 dating, and again, if you go to the exhibition and you'll see the panels talking about this, the dates are actually slightly too early for Queen Emma's death in 1052. But because she has these high nitrogen levels that seem to show up in the bone, it could be that she's eaten a lot of fish during her lifetime. And when individuals have eaten lots of fish, particularly marine fish as well, something can happen that we kind of term the fish effect. So because the carbon environment for marine organisms is more complex than that terrestrially, it actually can make the carbon-14 dates look slightly earlier than they truly are. So it can make them look slightly different. So that could be an explanation for that. But again, you know, research is ongoing. And we're currently looking at analysing kind of fish bones and other animal bones from Winchester to kind of get the kind of normal kind of levels that we would expect to see from material of this age. So again, I'm going to leave that with a kind of question mark of, you know, could this be Queen Emma at the moment? And more work is going on there. And again, the 3D prints, we did some really nice 3D prints of the remains. So none of the human remains have gone on display. They remain within the mortuary chests, but we have some nice 3D printed examples on display. Okay, so moving on to project 2, just briefly, because to bring in talking about another young individual. So I mentioned that I worked on the excavation of the Priory of St. Peter and St. Paul in Taunton. This was excavated way back in 2005. But again, I worked on this collection as part of my PhD research, and I still am doing various research on this collection at the moment. So in terms of the date of this cemetery site, so it's founded in the early 12th century and is utilised all the way up until the dissolution of the monasteries in 1539. The only part of real good dating evidence that we found on the site was this coin that's shown here in the image, which was a coin of Edward III, so dating to the mid-14th century. But more recently, so as I mentioned, I'm still doing research on this collection. And just last year, we did some radiocarbon dating, and we've had various dates, but again, spanning that kind of period, showing that the burials were going on kind of from that kind of early 12th up until, in this case, the 1450s was one of the latest dates that we got. Okay, and so yeah, with my PhD, I focused mainly on the juveniles, the children from this site. And that was because when we were excavating, we had about 50% of the skeletons were immature, and it seemed like quite a high number of young kind of skeletons, young individuals that we were finding. And what I looked at pathology in general. I particularly looked at evidence for stress to these children as they were growing. So evidence for malnutrition, vitamin deficiencies, and also for infectious diseases. And what I found was that status, so whether you had a high status burial in the church or whether you were out on the edges of the cemetery, didn't seem to have that much effect, but in fact all the children were quite stressed, even the high status ones. They showed just as much stress to them as the lower status individuals. So I just picked this individual to kind of briefly mention. I mentioned tuberculosis that we saw in one of my 19th century collections that I'm working on. But this individual here from the medieval period, we also have sign of tuberculosis in a young child, so age between 3 to 5 years. So I mentioned that we always look for any kind of indications of pathology. So essentially, when we're looking for pathology, we know what the normal skeleton should look like, and we know what normal variation looks like. So anything that looks unusual, abnormal, we kind of pick that up, we kind of investigate, and we try and work out what could be causing those particular lesions. So in this case, we've got an image on the left of some of the rib bones. And where the little white arrows are, you can see some kind of scooped out lesions. So these are destructive lesions. They're destroying the bone. We also have an image on the right of the skull. So we're looking at the right parietal bone and part of the occipital bone around the back of the skull. And there's a large destructive circular lesion towards the back of the skull. So when we see things like this, we obviously go and look at our, paleopathological textbooks. We look at the clinical literature as well, because that can be really useful. And these are quite typical lesions that you see on young children with tuberculosis. So I wrote a paper on this back in 2012 now, I think, suggesting that this was tuberculosis. More recently, I've been working with Michael Taylor at the University of Surrey. What he does, he takes samples and he does DNA analysis, But this time he's not looking at the DNA of the human themselves, he's actually looking for the DNA of the bacteria. So in this case, the Mycobacterium tuberculosis. And so he took some samples, he's been looking to see if this is tuberculosis, and it's kind of slightly hot off the press at the moment, but it looks like my diagnosis from the morphological lesions does appear to be correct, because he has found evidence for the aDNA of that bacteria. So again, that's another way that we can bring kind of in these scientific techniques to marry in with the morphological analysis that I undertake. Okay, and then on to my final site, just to talk to you briefly about. So this is the final project that I'm working on, which is actually several sites across Bristol. So when I was working at the University of Bristol, I started working on a site which is the St. Augustine's Chapel and St. Catherine's Convent site. Then another site was excavated by Avon Archaeology, who I work in quite close contact with, just down the road at St. George's Church. And there is actually another third site now, which I haven't got on this slide, but at the moment, lots of Victorian burials seem to be being excavated in Bristol. So hopefully I'm going to do lots of analysis on lots of these remains, and it's going to start to tell us a really interesting story about people of Bristol in this period. But I just picked, I think, 3 individuals just to mention. And I kind of picked them because they're either related to trauma or also related to surgical interventions, which again probably are related to trauma. So thinking about kind of the advent of surgery in kind of the 19th century Bristol, and how that was being undertaken at the Bristol Royal Infirmary as well. So to start off with, I quite like some examples that have been coming out that show trauma that suggests interpersonal violence. So on the image here on the left, we're looking at the skull. And if in particular you look at the nasal bones, hopefully you can see there's kind of quite a large gap, a large hole, but in the center of them, they're a bit kind of distorted. So essentially this individual has fractured their nasal bones. They've healed quite nicely. This individual also has a fracture to the distal left ulna. And I have another individual, skeleton 22, who also has a nasal fracture and also a fracture to the 5th metacarpal, which is the bone inside the hand. And these are quite classic fractures that you get when people are fist fighting. So essentially what we call pugilism. And there's quite a nice quote here from a Bristol newspaper from the early 19th century describing pugilism, so this bare knuckle fighting, as an elegant and fashionable science. And it was something that was very popular in Bristol at the time. And in fact, it wasn't just the men that were involved either. There's an image here of a famous Bristol boxer called James Belcher, but there's also in another newspaper article a story of his sister having a 50-minute fight in the streets of Bristol, kind of being urged on by her mother. So, you know, the women were getting in on the act as well. And then just to mention some surgical procedures, which are also really interesting from this collection. So here we have some evidence for a couple of amputations. So if we look at the image on the left-hand side, we're looking at this area here. So we're looking at the shoulder joint and the humerus, so this upper arm bone, that has been cut through. So hopefully you can see there's a really nice kind of clean cut coming through that bone. There's a little bit at the end where it seems to have snapped off before the saw has gone straight through. This is from a female. She's quite young. There's no evidence for healing on this bone. So that means that she didn't survive either the surgical procedure, she may have died on the operating table, or if she did survive for a little while, she didn't survive very long, because as soon as we have trauma to the body, to the bones, they start to heal themselves. They start to kind of put new bone growth on. So she hasn't survived this operation. In contrast, we have a male who's had his left leg amputated, and he has survived because his femur has really nicely healed over. All the bone has kind of nicely healed over. And in fact, when that individual was excavated, there were the remains of a wooden prosthetic leg that had been recovered with him, and also the buckle which held it on. So we know that he was surviving and getting around on his prosthetic, a bit like the image of the gentleman that I got from Bristol Libraries at the bottom of the slide there. So one of these individuals survived and the other one sadly didn't. Oh, and that's just him laid out full in the lab, again showing that, you know, he's got the one good leg and he's got the one leg that's been amputated. His good leg, I say it's a good leg, it does actually also show some trauma to the lower leg, so to the fibula, the smaller bone in the lower leg, but this has healed. So it may be that the trauma also affected the left leg, but was more extreme, and that the leg had to be amputated. And then my final individual that I just wanted to mention to you, because this might tell us something about kind of students learning and surgeons practicing at the BRI in Bristol. So we have at the site of St George's Cemetery, we had quite a lot of evidence for post-mortem autopsies. So we had craniotomies, so where they're taking off the top of the skull so they can access the brain. We see that here on this individual. So the top of the skull has been removed. Quite a lot of them are quite clumsy. They don't look particularly professional. So it makes me think maybe we're seeing some evidence of people who aren't that proficient at this yet. So maybe we're getting practice. But this individual in particular was interesting because here we have evidence for another amputation. So in the lower right image, we have the tibia and the fibula, so the lower leg bones. And hopefully you can see they've been sawn through kind of quite neatly. There's a neat cut through there. But in those other individuals that I showed you, we don't have the bit that's been removed buried with them. Whereas with this individual, the leg that's been sawn off and the foot as well has all been placed back with that individual and buried with them. And that means we can see evidence for what was wrong with that leg. So why were they amputating? And in this case, there doesn't actually seem to be anything wrong with it at all. So I've wondered whether they're doing this kind of post-mortem autopsy on this individual, and therefore at the same time, they may be getting in a bit of practice, because you don't really want to be chopping someone's leg off, amputating someone's leg if you haven't practiced before on a cadaver. So this may be evidence for kind of practice at the BRI. And the other image I've got down there just in the bottom left shows looking at the teeth. Hopefully you can see there's this kind of notch, this circular hole in the dentition. That tells me he was a smoker. So this is where he had his clay pipe and he was clenching it in his teeth and probably keeping it there all the time. And he actually also died at quite a young age. So he was aged between 25 to 35. So yeah, maybe not a good idea to be a smoker, he tells us. But I think that is where I'm going to stop.